Hi, everybody. I unfortunately am not able to be with you today. My son has been sick the entire weekend. And so I am going to try to do my best to actually bring you a lesson um, during your class period today and utilize the packet that you got last week as well as um, an article from Khan Academy to try and talk you through meiosis so that we don't lose time, right? When I come back, which hopefully will be on Tuesday, I will pass back quizzes to people who have actually taken them. For people who still need to take them, please try to be ready to go on Tuesday to do that. And I will put you out in the pod and have you take it. In your respective class periods, and we can kind of go from there. We can talk about um, the quiz that more than likely we will have next week, as well as kind of finishing up with some inheritance patterns and some Punnett squares. All right. Hopefully, everybody had a really nice weekend. But in both classes, kind of before we went to the weekend, one of the things that I asked you to do was to go through and actually read these 2 pages on meiosis. So they look like this in your packet, and this is what you're going to want to find. And then I ask you to actually do these matching. Okay. So a little bit later in this particular video, I will show you the answers for those. Also, to know that anything that goes through prometaphase 1, which is answer C, and prometaphase 2, which is letter I would not ask you because those are phases that we are not going to worry about. Okay, so they are definitely in your packet. There are pictures of them, but like I told you last week, whether it is in mitosis and or meiosis, we will not be doing those. All right. So when we left at the end of last week, okay, we had talked about the differences. Yikes, if I could get this to go correctly. Sorry. Um, we had talked about some differences between mitosis and meiosis. So to kind of pull that back and list those out for you, and you can feel free to obviously stop this video anytime you want to, write down things, make notes. I would highly recommend that. It definitely helps. Mitosis, as you may have actually seen last week, is a little bit difficult, and meiosis is going to have a lot more that goes with it. And so you're going to definitely want to be able to keep these two processes straight. Okay, so things that are different between them. One, mitosis actually is the process that we use to copy body cells. Okay, and meiosis is the process that we use to copy or have go through cell division for sex cells, so either egg cells or sperm cells. Mitosis takes this starting cell, which they actually call the mother cell, and it produces 2 cells. Okay. And these 2 cells are identical to each other, and each cell has 46 chromosomes in it. Okay. So that is actually kind of 2 differences. So mitosis takes a mom cell, produces 2 identical cells. Mitosis takes a mom cell with 46 chromosomes in it, produces 2 cells that each have 46 chromosomes in them as well. Because remember, when this is an interphase, it is going to make a copy. So it goes from 46 to 92, and then those 92 chromosomes split into each of the 2 cells, giving 46 to both of them. All right. Meiosis, on the other hand, takes a mother cell and produces 4 cells. Okay. They are not identical to one another. All right. As it tells you over here. And each of these 4 cells have 23 chromosomes in them, as opposed to the 46 that we actually get from mitosis. Okay. And the reason that it needs to be 23 rather than 46 is that because we are going to use the meiosis process to produce either 4 sperm cells or to produce 4 egg cells. All right. And then eventually, those egg cells and those sperm cells have the opportunity to potentially go through fertilization and produce what is known as a zygote. Okay, so the zygote is the first cell of any living organism. All right, and it's spelled ZY GOTE. So we need 23 chromosomes in all of dad's sperm cells. We need 23 chromosomes in all of mom's egg cells so that when they actually come together in the process of fertilization, we have the joining of the 23 chromosomes from each kind of party. And then 23 plus 23 would give us 46 chromosomes in that first zygote cell, which would be much like the mother cell. And then it would automatically start going through the cell cycle, which would start with interphase and then go through G1, S, G2, and then on to mitosis to produce two cells. And that is where the growing of a brand new individual would actually start. The other thing that we talked about is the academic vocabulary that goes with the numbers 46 and 23. So as we can kind of see over here on the left-hand side, all right, that when we actually talk about 46 chromosomes, or having the idea that we actually have pairs of similar chromosomes, okay, so it would be the same trait, not necessarily what we would call the same allele or the same piece of information. So if we were looking at diploid chromosomes, okay, then we would have chromosomes from mom and chromosomes from dad. And if we were going to go to, like, let's say the 13th chromosome, all right, and that just randomly happened to have information about eye color on it, okay, so both mom and dad's chromosome 13 would have information about eye color, but mom's chromosome might say blue eyes and dad's chromosomes might say brown eyes, all right, or green or hazel or any one of the number of choices that we have. But as long as we have information from both chromosomes, we get a diploid number. And in our case, that would be 46. The haploid number, all right, is half of that, meaning that it's only coming from one parent. And that is what meiosis will actually get us. So in our two divisions, okay, which is another difference that we'll talk about here in a minute. But when we get through the two divisions of meiosis, when we get these 4 haploid cells, they only have information coming from one parent's or one side's chromosomes, which is why there's only 23 of them. All right. So, and then one of the last differences that we actually have is that in mitosis, you have one division. So there's one cell division that actually takes place to get those two cells. In meiosis, the first cell division will give you 2 cells, and then in the second division, it will go from 2 cells to 4 cells. Okay. And then the only other difference that they actually have is this idea that mitosis does not have its chromosomes travel in what we call homologous pairs, but meiosis does. And so what a homologous pair or a homologous pair is, okay, is exactly what they are showing you, right down here at the bottom of the page. Okay, so like it says, homologous pair chromosomes are not identical, okay, although they could be. Okay, so if I went back to that chromosome 13 from mom and dad, all right, and they both had information for blue eyes or they both had information for brown eyes, then they would have the same trait and they would have the same allele. Okay, and allele is just whether or not it's a dominant trait or whether it is a recessive trait. So dominant, okay, more common. We are much more likely to actually see that genotype that's then expressed as a phenotype. So most of the time you see that as a capital letter, versus blue eyes that would actually be a recessive trait, right? And so the genotype for that would be a lowercase letter that comes from each parent because it is less likely to be present within within a given population. Okay, so homologous pairs can be identical. They don't have to be. They're very similar, like it says, in length and then what DNA information they actually happen to contain. Okay, so you can have long homologous pairs or tall homologous pairs, and you can also have short homologous pairs. Okay, so Prophase 1, or in the process of meiosis 1, what is different between it and the process of mitosis is that when it comes out of the cell cycle, so it's coming out of G1, S, G2, and now it's ready to go into meiosis. Okay. When we talked about mitosis actually happening for prophase, we talked about the chromatid, or like the chromatin actually kind of supercoiling into chromatids. And then they paired up like to actually make a chromosome. Okay. Here, they're going to do the exact same thing, but they're not going to stop once they actually have a chromosome. They're going to go one step further and they're actually going to pair up with their homologous pair. So their other chromosome that they actually match up with, which is why if we go back up to this, okay, so you have mitosis and meiosis that actually happen to be there. Okay. But in this one division, you see that all we're actually doing is separating the chromosomes here into chromatids, okay, in mitosis. And here they stay with their pair, okay? So they're going to hang out with the tall ones are going to hang out together. The small ones are going to hang out together. They did that up there in mitosis. It is just random in mitosis. They would not actually get together in homologous pairs there. Okay. But you can see that they stay together as homologous pairs until the very end here. Okay. So, all right. The other thing to actually know is that sex cells, egg cells and sperm cells, or sperm cells and oocytes can also just generically be called gametes. All right. And then they also talk to you about the idea that the gametes are haploid, which we already went through. Okay, so let's go ahead. That's kind of the review through things. I know first period had gotten kind of all the way through that. Third hour, you guys needed a little bit about the homologous pairs, gametes, all, and I think those were the two things that we hadn't done. And we're going to talk about crossing over as well. All right, so when we talk about meiosis, we talk about meiosis in terms of meiosis 1, which is the first cell division, or meiosis 2, which is the second cell division. Okay, so at the beginning in each one of these, it's going to give you like a little picture that goes along with it. And I'm also going to start to use the Khan Academy articles so that you can kind of see them because they do them in color. Here, they want you to color everything. And if you would like to actually color things as you're going along with them, that's fine. I feel like in first hour, you're going to have an easier time with that because there are colored pencils, all sorts of different ones that are in that kind of glass cabinet. That is just to, if you're looking at where the screen is in that room, they're going to be to that glass cabinet that's on the left-hand side. About 3/4 of the way down, there's all the stacks of colored pencils, and you guys could actually get those out. In room 115, it's going to be a little bit harder because I think that there are bags of colored pencils and bags of markers. They're over across from where Nolan and Cooper actually sit. So, like that other side of the table that runs right up against the screen is there, and I think there might be some that are actually sitting behind there. Okay, so you may want to color. You may just kind of want to listen today and then work potentially on coloring and going farther with things on Tuesday once I get back. And then I can also answer any questions that we have. Okay, so let's actually go ahead and get started. So like it says right down here, at the beginning of meiosis 1, the mother cell, or the cell that you actually start with, is an interphase. Okay. Now, that cell is going to be just like the cell that is in mitosis. So it is going to go through a cell cycle that is the exact same as the cell cycle for mitosis. The only thing that changes is the M phase. If we're copying body cells, the M phase is mitosis, prophase, metaphase, anaphase, telophase, and then cytokinesis, where the cell splits, or PMAT-C. Here, The cell cycle is going to be interphase, G1, S, G2, and then the M phase is going to be meiosis. All right. So even though they don't show you it here, okay, the mother cell is actually going to be in interphase, right? And I'm going to scroll down in this. I will link this. Well, I don't necessarily need to link the article because you'll actually have it here, but I could link the article in our cell cycle mitosis meiosis folder. So if you'd like it by itself, you definitely can have it. Okay, so we're going to skip this part for a little bit. Okay, it doesn't give you an interface here. Okay, but you have an interface picture from your mitosis coloring page. Okay, so let's go back and just kind of review real quick. Okay, a little bit about what interface is actually going to do. So in G1, which is gap 1, okay, all of your organelles are actually going to copy. All of your molecular building blocks that you need for other things are going to copy. The DNA is in chromatin phase, so it is going to look like those cooked spaghetti noodles. Okay. Then it is going to go into S phase or synthesis phase. And in that synthesis phase, it is going to copy DNA. So DNA replication is going to happen. A reminder for that, even though I don't have it on this video, okay, that the DNA replication process is semi-conservative. What we mean by that is that it is going to go from one strand to two identical strands, but each half of the strand comes from a different source. All right. So half of each of the strands is going to come from your original DNA template or the old DNA. The new DNA is going to be formed in one of two ways. Okay, one side that actually goes towards the replication fork. Okay, so or towards like where the two strands are actually going to join, like the point in the Y. If it is going towards that replication fork, it is called the leading strand. Okay, now the leading strand is going to be adding nucleotides. Okay, once helicase actually breaks it open and separates it so that the hydrogen forces between the nitrogen bases are pulled apart, then the DNA polymerase is going to come in and it is going to add those nitrogen bases one at a time in a continuous fashion. All right. While the other strand, which is going away from that kind of like the point in the middle where the two strands are going to join, okay, and it's moving out towards the end of the DNA strand, that is called the lagging strand. Okay, so same thing, helicase has opened it, you have a DNA polymerase that's there. On both the leading strand and the lagging strand, you do have to have a primer to tell the DNA polymerase to like do something, but we're really not worrying about the primer. All right. So then the lagging strand attaches nucleotides together in segments or in what we call the Okazaki fragments. But it is a section or a fragment at a time until it actually gets to the end of the area where you are copying it. Okay, so that's the S phase, kind of a little review from last week. Might be helpful to review that given your quiz. Okay, and then after the S phase, we have the G2 phase, which is gap 2. Okay, it is going to go through and it is going to, that is where protein synthesis is going to happen. All right. So if we have an error in DNA, the DNA polymerase and DNA ligase did not catch, okay, because DNA ligase actually like proofreads things, seals up everything, and kind of puts it all back together at the end of DNA replication. Then there are errors in there. And you guys all know about the errors that you have based on the mutations that are there. All right. So protein synthesis is going to happen and the cell is going to prepare for, in this case, meiosis. Okay. So what happens in prophase 1? Okay. So prophase 1 is very similar to the prophase that actually happened in mitosis. Okay. So as we can actually see here, all right, we have gone from chromatin. Okay. So the chromatin that was actually like wet spaghetti noodles in interphase has coiled up to actually form a chromatid. It is joined together with its sister chromatid to actually make a chromosome held together in the center, okay, by all of these little things, okay, where the P section would be on top because it's shorter and our Q sections would be on the bottom because they are longer. That is called a centromere. And so it makes a chromosome. And then the new part, okay, so right here it says something very unique and different happens. The additional step in prophase 1 for meiosis is that this is where your homologous chromosomes or your homologous pairs are actually going to pair up. So the chromosome 1 from mom would find the chromosome 1 from dad, and they would actually pair up. Okay, and remember that when you do karyotypes, that your big chromosomes, or like chromosome 1, 2, 3, and so on, are going to be much longer than the chromosomes that you actually get as you head towards chromosome 22, if you're looking at body cells, and then also chromosomes 23, which determine gender. Okay, so those are all going to actually join together. Okay, so technically what it's going to look like is 4 chromatids. Okay, and sometimes depending on what source you're actually looking at, okay, they can call that a tetrad. Okay, so if you've come across that term somewhere and you're like, what in the heck is that? It's just a way to actually say that there are 4, 1, 2, 3, 4 chromatids that are making up the 2 homologous pairs. Okay, so it doesn't matter what it is, you're always going to see 4 of them. Okay, so this part where they actually stick together, where they talk about synapses, we are not going to do. Okay, now once they are lined up in their homologous pairs, okay, they do this process between sister chromatids, okay, that is called crossing over. All right, so I'm going to go back over here, all right, and I'm going to show you what this actually looks like. All right, so if we are looking at prophase 1 on this particular article from Khan Academy, you can see that here they are showing you that you have the chromatin that looks like spaghetti noodles that is starting to actually coil up, right? So that it will end up looking like this. So it'll end up looking like a chromosome. So chromatid, chromatid, sister chromatids held together by a centromere to make a chromosome. And then the new part is that it finds its buddy, if you will, so its homologous pair. The other 2 things that happen that are the exact same in prophase 1 is that nuclear envelope or that nuclear membrane is going to start to actually dissolve or go away. And then here are our centrioles, right? And they're going to start to move to opposite ends and they're going to form that mitotic spindle. Okay, so many of you talked about that in your quiz and did a great job with those, but those are now we actually have more things that actually happen in prophase 1. Okay, now in prophase 1, they just said that they were talking about this idea that these sister chromatids, okay, when they line up between sister chromatids between the mom chromosome and the dad chromosome, and it doesn't really matter how they line up. Okay, so you can see like on some sides, you might get like if the darker one were the dad chromosome and or like this kind of bluish purple one was the dad chromosome and this red one, reddish one was the mom chromosome. You can see that on this side it would be mom dad and on this one it's mom dad. Okay, there's no rhyme or reason to how they actually line up. Okay, so it could be like mom, 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 dad, mom, dad, mom, dad, 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 mom, mom, and it changes every time when they actually line up. Okay, so crossing over, I did a little bit with in first hour. I didn't touch it in third hour only because third hour had a lot more questions about the quiz last week than first hour did. So we didn't get to it, which is fine. Okay, so here's what crossing over actually looks like. Okay, these are your four chromatids. Okay, so if we were taking this and we were saying this is a mom's chromosome, whatever number you actually want it to be. Okay, one of the things I would actually recommend, okay, is that you could tag a chromosome or you could tag your chromosome with your error in it in the pictures that you actually do, because then you can kind of see how it gets copied and how it moves from one cell to another. All right. So if my disorder was on chromosome 6, okay, and I know that they're all over. You can pick whichever one you wanted, or it's on the X chromosome. Okay. So here's mom's information, and it's identical. So mom has, in this example, has dominant genes for gene A, B, and C. Okay. Now, if you actually have one of those, you could say gene B is the gene that your genetic disorder is on. So if I have the HBB gene or the hexa gene or the OPN1, either SW, MW, or LW gene, or I have INS or any one of a number of genes that are on there, you could say this is where the information actually happens to be. Okay. And then the same thing for dad. Okay. Except dad's chromosome in this particular example is all recessive information. Okay. So neither dominant nor recessive is mutated. It's a different trait. Okay. So, there's nothing potentially that actually goes wrong in those. But in somebody who actually has a genetic disorder, you could say it's a dominant trait, like it's an autosomal dominant trait or it's an autosomal recessive trait. If it's on the X chromosome, you could say that it's a sex linked dominant trait or it's a sex linked recessive trait. Although most of the time we just say it's a sex linked trait. Okay. And then here's what actually happens. Okay. Mutations add a bunch of different variety, and it's one way that actually adds variety to the genetic, like what we call the gene pool. Okay, so it's a variety and it makes us look different, and so that we're not all exactly the same and clones of one another. And I talked about that, like being weird and boring and all of those kind of things. So like even if we had different personalities and that because of our environment and that, we would still all look the same and it wouldn't be exciting. Mutations are one source of variety. Crossing over is the other big source of variety. So what ends up happening is you have sister chromatids that now are coming from two different individuals. Okay. So in this case, like I said, I just randomly chose the red one to actually be the mom and the purple one to be the dad, but you could easily choose the other. It doesn't make any difference. And they switch information. Okay. And that's what it's called crossing over, because the sister chromatids are going to cross over. Now, in this particular one, they just showed you that the information that is on section C crossed. And that's fine because now it's different. Every single one of these chromatids now has different information on it. But it doesn't just have to switch C. This time it switched C. The next time it actually goes through, it could switch A and C. It could switch just B. It could switch just A. It could switch all of them. It is random every single time the process of crossing over happens in meiosis 1. Okay. So when you go to look at them and in your packet, you have crossing over pages that you can actually color. It's going to show you something that looks like that. Okay. So now, okay, it also comes down and it talks about this like complex. We're not going to do the complex. Okay. Um, there's enough vocabulary that actually happens with this, and you can get caught up in the vocab, which is already crazy. We don't need to actually do more of that. Okay. Now, so that's prophase 1. All right. And we'll go back a little bit for this diagram. I'm sorry if you can hear my cat in the background. So I actually shut the door so that no one would bother me. And he is used to being in here with me the whole time. So I apologize for that. Metaphase 1, again, means middle. And so you are going to see the chromosomes all lined up in the middle. The only difference between metaphase 1 in meiosis 1 and metaphase in mitosis is that they are just lined up with their homologous pair. All right. So if we look at kind of metaphase and anaphase, like they talk to you about it, that they're splitting it apart. Okay. That here, when you actually look at anaphase, and this one is a little bit different and is somewhat misleading. You have to look really carefully at it because it kind of looks like it's only separating one of them, but there's actually two of them that are coming through there. So this is a much better picture off of the Khan Academy one to kind of look at. That if you look at metaphase, okay, and sometimes they can actually say, it does say that crossing over is here. They don't show you the crossing over. I guess it kind of actually is showing you where the overlap is. Here we go. All right. In metaphase 1, where they're actually all lined up. Okay. And then in anaphase, rather than the chromatids actually pulling apart from each other, they have the chromosomes, like your homologous pairs are going to separate from one another. And so just like an anaphase in mitosis, you have these you have them starting to move away from each other. So anaphase still means away. All right. But this time the sister chromatids are actually going to stay together. Okay. But they're just going to move to opposite ends of the cell. Okay. And then in telophase 1, much like you actually saw in mitosis, you're going to see the same thing start to happen. All right. Now you have each of these chromosomes. They are not going to be actively dividing. So you start to see them unravel and they go from being kind of your like in chromatid form that makes a chromosome into chromatin. Okay. And so you can kind of see how they go from being thick here to thin here on both sides as you get kind of two not identical cells, though. This time they do not look identical. All right. You do see that the nuclear membrane or the nuclear envelope is starting to reform, and that your centrioles have pulled in their mitotic spindle. Okay. They don't need it right now. Okay. So when we look at this, okay, that is kind of where we are. And when you get to kind of the end of telophase 1, your two cells are going to separate and you're going to have cytokinesis occur. So just like we did in mitosis, the cytoplasm is going to push in from either side, and you are going to get two kind of two separate cells. Okay. So this is going to talk you through kind of everything that we just looked at in pictures. Okay. And now it shows us everything doubled, okay, because we had two cells. Now there is what they call interkinesis, all right, that happens between cytokinesis and prophase 2, okay. It is not an interphase, okay. It's kind of just a break or a pause between the first cell division in meiosis and the second cell division in meiosis. Okay. But it is not like an interphase where you go through G1S and G2 again. You don't need to copy anything. It's pretty much ready to go. Now, here, okay, this looks very much like mitosis. Okay. In fact, like it actually says in the article up above, it says here it is, we have mitosis, and they call it mitosis for haploid cells because it's mitosis, but your end game here, okay, is not to produce identical diploid cells. Your result here is going to be to produce 4 non-identical cells that have the haploid number of chromosomes. They have 23. So prophase 2, all right, looks just the same as the prophase in mitosis does. All right, where we have our chromatin, and it is now going to actually start to go from chromatin to a chromosome. They are um, they are already kind of in chromosome form. Okay. They don't kind of detach and then reattach to form a chromosome here. All right. But they do kind of unwind a little bit. All right. But they do stay connected to their sister chromatids. You'll see that because they're going to actively divide again, that nuclear envelope or membrane is going to start to dissolve or disappear. And then your centrioles are starting to move to opposite ends again. They're going to produce that mitotic spindle, that protein that's going to come out of their cylinders to hold on to the chromosomes. All right. Metaphase 2, all right, is right here. And I like it being bigger, but it's hard to actually see the tags for the stages. So here in metaphase 2, same thing. They're lining up in the middle. This time it's just the chromosome. So you'll notice that it's not with its homologous pair because its homologous pair is in another cell entirely now. All right. Anaphase this time looks like anaphase 2 in meiosis 2 looks like anaphase from meiosis. The chromosomes will split at their centromeres, that part that actually holds them together, and they will separate and they will move to opposite ends of their cell. All right. And then at the very end, where we have telophase, all right, you will notice that when you look at it in every single cell, because you're getting 4 cells, that none of them are identical to each other. Okay. So every single one that you have is different. Okay. But the only thing that's really the same is that they all have 23 chromosomes or the haploid number of chromosomes. Okay. They are going to unwind back into chromatin. Okay. And the nuclear membrane or the nuclear envelope is reforming and the mitotic spindle is no longer there. Okay. So the centrioles are kind of, there's one centriole now in each cell. Okay. And when cytokinesis actually happens twice this time, it's going to push in on the cytoplasm here to give us 2 cells here. And then it's going to push in on these 2 cells to give us another 2 cells here. All right. And so we will end up with 4 cells from the mother cell that we started with, not identical, each one of them having 23 chromosomes or the haploid number, all right, within each cell. Okay. And this particular handout that you have in your packet kind of talks you through each one of those. Again, it does mention prometaphase in each thing, and we're not going to do prometaphase, okay? But it does show you them lining up and splitting, and then you actually ending up getting 4 gametes, okay? Now, those gametes, or the sex cells that you actually end up with, okay, if you are a male, or we're talking about males, okay, then those 4 cells are going to go through a process that's called spermatogenesis. And I'm going to talk to you about that in a separate video here in a moment, okay, about how we actually take these gametes that are at the end of meiosis 2 and how they become sperm cells, okay? Or if we're talking about females and we're at the end of meiosis 2, how these 4 gametes leave the end of meiosis 2, okay, and then go to actually form 4 egg cells, okay? Because they don't go back and actually start the cell cycle all over again like mitosis does, okay? So something entirely different actually happens to them. Okay, so that actually gives us that for your meiosis questions. Okay, for these, I want to go through the answers with you, and I'm going to end this particular video, and then the next video that you will be looking at, okay, is the one that talks about how we take either the 4 gametes and make them sperm cells, or we take 4 gametes and have them develop into egg cells. Okay, so for your meiosis questions, okay, these are all the blank ones. Okay, and then let's go back and actually look at the answer key so that you have this. Chromatin is actually visible during interphase, right? That's our kind of wet spaghetti noodle or cooked spaghetti noodle that we would have. All right. For 2 and 3, 2 and 3 are both prophase 1. Okay, so the only one that I would ask you about is actually number 2. Like if I were talking about where chromosomes are actually first visible, it would be in prophase 1. Like I said, we're not going to do synapsis, so that one probably isn't one that I would expect you to know. All right. If we go down to number 4, it says during this phase, replicated or copied chromosomes become visible, but they're not sticking together in pairs. Okay. That would be prophase 2. And they're not going to be together in pairs because the homologous pairs have already split at that time. So now we only have 1 chromosome from each one in each cell that we have. Okay, number 5 is not going to be applicable. You can mark that one out because I'm not going to have you be responsible for the prometaphase phase in either mitosis or meiosis, so that one would not be appropriate to ask you. Number 6, where it says, neither would number 6 because that's prometaphase 2, so we're not going to do that one. 7 says, at this point in time, homologous pairs of chromosomes are aligned in the center of the cell. That's metaphase 1. Okay, the key part being that if they're with their homologous pair, it's going to be during the first cell division. If they're by themselves, just as regular chromosomes, it'll be in the second cell division. Okay, number 8 moves us to metaphase 2, where it says, at this point in time, replicated chromosomes, not pairs, are aligned in the center of the cell. So center of the cell lets us know that it's metaphase. Your key points are whether they're in homologous pairs or they're just chromosomes. Okay, number 9 is homologous replicated chromosomes in a pair split into separated replicated chromosomes and begin moving towards the plate. Because they are moving towards the poles or opposite ends of the equatorial plate or the middle of the cell, that is anaphase. But because they are in homologous pairs, it's anaphase 1. All right. And then 10, replicated chromosomes split into separate chromosomes and begin moving towards the pole. It'd probably even be better if they said replicated chromosomes split into separate chromatids, but they didn't, which is fine. It's anaphase 2. Really what it's getting you to realize is whether the wording behind whether it's the first division or the second division. That's also going to be important for you to know when you are actually explaining it to either the person who has a genetic disorder or the parents of the person who has it. And so this is just good practice for that. F says replicated chromosomes finish moving towards the poles in this phase. And F is going to be telophase 1. And then they come down and they say chromosomes not replicated, okay, because they're not copied. They've already been copied, so they don't need to copy again. Finish moving towards the poles in this phase would be telophase 2. Okay. And then 13, for G, scientists use this word to refer kind of as a pause between meiosis 1 and mitosis 2. During this phase, centrosomes are going to replicate, but chromosomes do not. Okay. I'm not even going to hold you responsible for that. If you just want to know that interkinesis is just a pause or kind of a, like this time frame that exists, it's kind of a resting phase between the two cell divisions, you're going to be good. Okay. And then 14 says, in this event, 2 cells split into the final 4 daughter cells would be cytokinesis. All right. And then these are the final products of meiosis, which would be gametes. Okay. So hopefully that answer key actually helps you. Okay. And you also know which ones you are actually responsible for. And that this video has kind of helped you look at how meiosis is different than mitosis and how meiosis 1 is different from meiosis 2 and how they're both kind of similar to meiosis, but also different. Okay. Thank you so much for those. If you can actually get through this video, that will be good for today. And if you have time, you can actually start looking at spermatogenesis and oogenesis for tomorrow. But this is about a class period, so you should be good to go.